Hello, in the previous class I showed you multi-image of a tissue sample, cancerous tissue sample. It was prostate cancer tissue sample, and there we showed how proteins can be imaged from a tissue surface. Now, in the last class also I told you that I will introduce you with another kind of imaging technology based on mass spectrometry platform. That is what we are going to discuss today. The ionization method itself has the name DESI, D-E-S-I, Desorption Electrospary Ionization. We know electrospray ionization, that is the first thing we discussed in this class, meaning in this course. And there is an added term, desorption. So something is getting desorbed by using electrospray ionization technique, and the experimental substance is a surface. It may be a biological tissue, it may be anything, it may be a glass slide with a thumb impression on it, it may be any forensic substance. So there, in this technological platform, it's not like the previous one. We are going to image proteins and peptides. Here, to be honest, it's not restricted on any molecule. Mostly it is focused on small molecules, but it can go to proteins level as well. So I am citing here the paper that the first came, that is the discovery of desorption electrospray ionization mass spectrometry based imaging technique, that was from Professor Graham Cook's laboratory, and that came in Science 2004, volume 306, page 471 to 473, I guess. So basically what it tells is, the basis of the technique is desorption electrospray ionization is carried out by directing electrosprayed charged droplets and ions of solvent onto the surface to be analyzed. As I said, the surface can be anything. The impact of the charged particle on the surface produces gaseous ions of the material originally present on the surface, and that finally resulting mass spectra is similar to the normal ESI mass spectra. So it's not like laser firing, it's a solvent jet that we are giving to the surface, experimental surface, and from there we are taking out analyte in the gas phase. So here the schematic representation of desorption electrospray ionization technique, where there is gas jet, and main important thing is this spray of solvents. And solvent is spraying on the surface on which our sample of interest is sitting, and then this solvent has charged ions in it, so it's causing the analyte from the experimental surface to come out of the surface into gas phase, and then obviously there is ionization of those analyte. So basically we are talking about another stage, another phase of ionization, which is secondary ions that are being thrown out, expelled out from the surface by the solvent jet, and then that is being taken inside mass spectrometer, so which is finally analyzed to give the electrospray, meaning ESI mass spectra. So this is the desorption comes from here, meaning from the surface, whichever was there on the surface, that is being dissolved. So that's why desorption. So desorption electrospray ionization technique, or DESI technique. Now let us see the principle in bit more detail. This is actually a housing system where DESI based experiment, Desi based imaging experiment is performed, and this can be hooked up with a mass spectrometer. So, if we focus on this area, which is the main key area of this technology. So, here it is the spray through which this spray happens, and then this is the place through which this is inlets basically. So, through which the molecular ions are taken into. Okay. So, this spray is this one and this inlet is here. Now, these distances roughly is mentioned like this here, D1, that is the distance between the surface and the spraying system is about 1 to 2 millimeter. D2, that is the distance between the emitter and inlet, the sprayer, emitter and inlet is about 3 to 4 millimeter, this distance, or rather this distance in the instrument itself. D3 is the distance of inlet from the surface, like this one, okay, or this one. So, it is about 0 to 1 millimeter, it is almost very close to the surface, so that we lose minimum ions that are being produced from the surface upon desorption. And the angle of spray, that is also a very important parameter, is maintained about 55 degree. So, here in this schematic also it's showing bit detail, the one that I showed previously in the first publication in the science paper of Graham Cook. Now, same picture here, so but more bit of elaborative form. So, these are primarily charged droplets that is coming out from the spray, and its high voltage is applied here about 5 kilovolt, and that is what we saw here in this picture. This high voltage is applied, and then these charged droplets coming and hitting on the surface, and then it is causing the ions from the analyte from the surface. These are the analyte on the surface. So, that comes out and with meaning being charged. So, those are secondary charged droplets, and then this is being taken into inlet. Inlet meaning inlet of the mass spectrometer. And obviously, there is a gas flow. So, nitrogen gas is the nebulizing gas. Okay. So, the key points about this technique is, here tissues are simply sectioned and images and imaged basically. No matrix is required for this technique. Like we have discussed in the previous class, multi-mass spectrometry based imaging. So, there we had to lay the matrix through spraying, but this is not required here. More suitable for small molecule like metabolites and lipids. Imaging can be generated in the pixel size of 80 to 100 micrometer. I think nowadays it is even lesser, but is 80 to 100 micron is pretty good in terms of dimension of the pixel. The ionization source is ionization source works at ambient pressure and temperature. Okay, this does not require any sample processing. So, the one that we observed in case of multi-mass spectrometry based imaging of tissues, where we had to remove salts and lipids from the surface first, so that it does not interfere with the ionization of the peptide, and for that we had to take the tissue through a series of solutions. One of them, the important, is Carnoy's solution, but no such processing is required at all. And the beauty is several thousands of distribution of maps or ion images can be formed, label-free mapping of many analytes, metabolites and proteins. We don't need to label it anymore, and simultaneous analysis of highly heterogeneous samples like biopsy tissues. Okay. So these are the advantages. Now I'll cite an example where this technology is used to image the biological tissue, prostate cancer, focusing at small molecules. Previously I showed you prostate cancer tissue imaging in terms of proteins. Today we will discuss about this prostate cancer tissue in terms of imaging of small molecules, metabolites. So prostate cancer is commonly diagnosed as solid organ cancer that we described in the previous classes, and is one of the leading cause of cancer death in men. We do have a biomarker, PSA, prostate specific antigen, that meaning quantification of prostate specific antigen actually helps people to diagnose rapidly or at the early stage, I should say, not rapid, but at the early stage of prostate cancer, when it is confined to the prostate tissue itself. It hasn't metastasized, so it hasn't gone to any other organ. And many of these localized prostate cancer are treated by surgically removing the entire prostate, which name is called prostatectomy. The presence of cancer cell at the age of surgical resection or surgical margin, that is called, or positive surgical margin, is associated with higher rate of recurrence and death from prostate cancer. This is very important. While removing prostate tissue, if there is cancerous tissues at the age where the surgeon is removing the tissue by knife, by cutting it, so if it is at the age, then there is a chance to leave some of the cells somewhere here and there, and that is dangerous because that actually causes recurrence of the cancer and ultimately leads to death in prostate cancer. Therefore, it is important clinical challenge in prostate cancer management is to devise a rapid and highly accurate method to detect cancerous cells in real time to allow resection of additional periprostatic tissues and reduce cancer recurrence after surgery. That is the main simple motto. Now, in DESI based mass spectrometry imaging, DESI-MSI, mass spectrometric imaging, that MSI stands for, can rapidly evaluate the tissue metabolome in situ by simultaneously characterizing hundreds of lipids and metabolites. In multi-based, we have talked about imaging of proteins. Here we are talking about imaging of lipids and metabolites mostly. Given the known alteration in metabolite pathway that is known from the literature, this DESI MSI of metabolites and lipids was explored to discriminate cancer from normal tissue obtained from radical prostatectomy. I am talking from this paper where authors actually made use of the known literature information, metabolic pathway of prostate cancer, and then focusing on that metabolites, how they are distributed across the tissues, both in case of cancerous tissues and normal tissues, and therefore tried to come to some conclusion of cancer related biomarker, which is our main topic of discussion today. So in DESI MSI, this is performed in negative ion mode. If we want to see lipids, best is negative ion mode, even small metabolites, negative ion mode, which are negatively charged species like glutamate, and then obviously I showed you at the tip of capillary there is huge voltage, about 5 kilovolt. M by Z range is 50 to 1000 because we are focusing mostly on small molecule. were no more interested in large molecules, and spatial resolution about 200 micrometer, the spray pod diameter. Okay. And using histopathology compatible solvent system, that is 1:1 volume by volume, dimethyl formamide and acetonitrile, DMF ACN 1:1 mixture, that is what is used to provide solvent jet on the tissue and a flow rate of 0.7 microliter per minute was maintained. Now, what is the basis of this? I will come to the to explain these tissues. The metabolite and lipid imaging, that is the main point that we are addressing here. Here this figure shows a representative negative ion mode Desi mass spectrum of a prostate tissue specimen in the range of 500 to 200, sorry 50 to 200, because not even we don't need to go till 1000. Metabolites 50 to 200, where most of small metabolites and glucose are detected. So, this is the mass spectra, is basically ESI mass spectra, though we got it from DESI platform. And this figure B shows the higher abundance of different metabolites across the tissues, like erythrose, glutamate, glucose, sorbitol. These are higher abundant in case of prostate tissues, and lower abundance of succinate, malate, salicylate, aconitate and citrate in cancerous tissues compared to the normal. So, these are the metabolites higher in concentration, and these are the metabolites lower in concentration compared to the normal tissue that is seen here. Now, in negative ion mode Desi mass spectrum, that is what we recorded here. I didn't record, obviously authors of this paper recorded here, and then a spatial distribution of 14 different small metabolites in prostate tissue specimen, that was highlighted. So, there are 14 metabolites, and this is H&E stained tissue, same tissue section. Okay. And then 14 different small metabolites in a prostate tissue specimen. So, upper left, that is this one, upper left corner, corresponding to H&E staining of the tissue sample, that contains both normal, that is this black outline, this is the normal tissue, and the red outline corresponds to cancerous zone. Okay. In B, some of the individuals metabolite distributions are mapped throughout the tissue, that is what I am showing it here. And in A, abundance of a small metabolites are displayed by averaging all pixels over a typical line of scan. So, this is basically, it is also showing relative abundance. Basically, it is a mass spectra. So, y axis obviously gives relative abundance, and x axis is m by z. So, these metabolites, whatever, those are higher in concentration, like lactate, succinate, erythrose, malate, glucose. So, you see this right side is cancerous zone. So, in these sides basically whatever metabolites are excess, that is labeled here, those are higher in concentration in cancerous region, whereas these like aconitate, this is more in normal, less in cancer. Okay. Like this one, malate, another metabolite, it is more in normal tissue rather than cancerous. Succinate, more in normal than cancerous, like that. Okay. Now, so based on this observation, what author did, they observed that it was clear that there was high level of glucose and low level of citrate in cancer. So, author attempted to use imaging of this glucose and citrate to characterize whether prostate tissue specimen was cancerous or normal. So, glucose and citrate that was taken as marker most likely, and we will see that actually they have taken a ratio of it. So, interestingly, the ratio of these two ions, glucose over citrate from the tissue specimen, and constructed image of that ratio, it was possible to differentiate prostate cancer from benign prostate tissue. Again, normal cancer, like here, this portion is representing inflammation, this portion is trauma, and black marking is representing normal tissue, red marking representing cancer one. So, distribution of glucose over citrate ratio of some representative prostate tissue specimens showing significant elevation of glucose over citrate ratio in cancer. The top of each panel, A to O, all these top panel, that is representing different tissues, H&E stain tissues, histopathological evaluation of the corresponding tissues, whereas cancerous areas have been demarcated by red, benign, and benign areas are demarcated by black, stroma region by green, like here stroma region, and here stroma region, here stroma region, and then inflammation area by blue. Okay. So, this is H&E stained tissue, same is this image, mass spec image tissue. This is H&E stained tissue, mass spec imaging tissue, and then glucose over citrate ratio is plotted basically here, and you see that red is the most intense part. Now, we can correlate very well wherever the cancer is populated, cancerous cell is populated, this glucose over citrate ratio is high. You see in all the tissues, wherever the cancer cells are more concentrated through histopathological based image, which is obviously gold standard, the ratio of glucose over citrate is high. Okay? And wherever these normal cells are there, then this ratio is no more high, this is less. So, this is very important information, that is what they are actually they have drawn conclusion from this observation. So, there is a distinct cutoff at the border between normal and malignant prostate tissues as determined by the glucose over citrate ratio. So, this is a tissue, for example. So, it has inflammation this portion, normal by black, encircled by black, and then cancer by red. Now, this represents citrate, and this represents glucose. So, extracted ion chromatogram of glucose and citrate over line scan. See this line is scan basically, and that contains on left side benign zone and right side cancerous zone, and H&E stain tissues actually showing the position of different tissues here. Now, this portion, this green line represents the concentration of citrate, and the pink one is representing glucose. So, if we look at this line that demarcates normal and cancer tissues, at that zone itself there is drastic change in the ratio. Okay. So, that is what was observed here. There is a distinct cutoff at the border between normal and malignant prostate tissues, which can be determined by the glucose over citrate ratio. Now, here in this plot, this ratio is been plotted for several normal subjects and cancers, and that was training set, this is validation set, again the same strategy, normal patients or normal tissues glucose over citrate ratio vis-a-vis cancerous tissues glucose over citrate ratio. So, what was the observation? There was a significant difference in the distribution of glucose over citrate ratio between prostate cancer and normal tissue, and the specimen could be accurately classified as cancer if the glucose over citrate ratio is more than 1, like entire this zone, and if the ratio is less than 0.5, mostly in this is 0.5, so this entire zone, this is 0.5, so this entire zone, then the tissue is normal. So this is very important issues, this is very important observation, that glucose over citrate ratio can act as a biomarker to define surgical margin very precisely, because if the borderline, that's this image is very important, borderline, at the borderline you see how the ratio is changing drastically from 1 to more than 1 in the cancerous zone to less than 0.5 in the normal zone. So therefore the conclusion is the measurement of glucose over citrate ion signal ratio accurately predicted cancer when this ratio exceeds 1 and normal prostate when this ratio is less than 0.5. So hopefully this strategy might be used someday in the diagnostics to detect surgical margin precisely, to define the surgical margin precisely, I should say, once this DESI based imaging is approved by the competent authority to use it in diagnostic purpose. So with that, I'll end the discussion on DESI based or mass spec based imaging of tissues, biological tissues. So I'll end the class here. Thank you.